Active transport is the transport of solute molecules that travel from lower concentrations to higher concentrations. There are three types of active transports in bacteria: primary active transport, secondary active transport, and group translocations. They differ in terms of energy used to drive transport and on whether or not the transported molecule is modified as it enters. Active transport and facilitated diffusion both involve carrier proteins, but facilitated diffusion does not use metabolic energy.
Primary active transport is mediated by carriers called primary active transporters. They use energy provided by ATP hydrolysis to move substances against a concentration gradient without modifying them. Primary active transports are uniporters, which means they move a single substance across the cellular membrane.
Secondary active transport couples the potential energy of ion gradients to transport of substance without modifying them. Secondary active transporters are co-transporters, meaning they move two substances simultaneously across the membrane. A co-transporter is made up of a symporter, which moves substances in the same direction, and an antiporter, which moves substances in an opposite direction. Secondary active transporters include the major facilitator superfamily proteins, MFS proteins. MFS transporters use ion gradients, some of which are created by bacteria during their metabolic process. For instance, electron transport during energy-conserving processes generates a proton gradient in which protons are at a higher concentration outside the cell than inside. The proton gradient can be used to do cellular work, including secondary active transport. The lactose permease of E. coli is a well-studied secondary active transporter. It is a single proton that transports a lactose molecule inward as a proton simultaneously enters the cell. This is an example of a symport. An example of primary active transport is ATP binding cassette transporters. In this example, ABC transporters are importing substances. Most ABC transporters consist of two hydrophobic membrane spanning domains associated on their cytoplasmic surfaces with two ATP binding domains. The membrane spanning domains form a pore in the membrane, and the ATP binding domains bind and hydrolyze ATP to drive uptake. ABC transporters employ substrate binding proteins. After binding solute, the solute binding proteins approaches the ABC transporter. Solute binding protein attaches to a transporter and releases solute. Energy released by the hydrolysis of ATP drives movement of solute across membrane.
Group translocation. In group translocation, a molecule that is chemically modified as it is brought into the cell. Best known group translocation system is the phosphophenol pyruvate sugar phosphorotransferase system, better known as PTS, which transports a variety of sugars while phosphorylating them using phosphophenol pyruvate, or PEP. PEP is used to synthesize ATP as the phosphate donor. Phosphorylase system is the transfer of phosphate from PEP to the incoming molecule involving several proteins. Phosphate is transferred from PEP to enzyme 2 with the help from enzyme 1 and the heat-stable protein. Enzyme 2 phosphorylates the sugar molecule as it